Hello, and welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines. People just like you, working to understand viruses and how they affect you. With the increased detection of variant of concern viruses, coupled with the slow pace of the vaccine rollout, on January 26, 2021, we talked with Dr. Tristan Jordan, a postdoc in the 10-over lab at the Icahn School of Medicine at Mount Sinai in New York City, about his work using CRISPR screens to identify new, potentially druggable targets for SARS-CoV-2 and the importance of vaccination, especially in people of color who have been hard hit by this pandemic. Tristan received his PhD from the University of Chicago, where he worked in the lab of Glenn Randall, focusing on understanding how dengue virus modulates cell signaling pathways to initiate a proviral lipid droplet-specific autophagy program. In the 10-over lab, he continues work on the cell biology of virus-host interactions, including SARS-CoV-2, and the regulation of the antiviral interferon response. Hi, Tristan. I'm happy to have you with us today. Why don't you tell us about yourself? How did you become interested in virology research?
Yeah, so I was trying to think about this. I don't know how I actually became interested, but somewhere along the way, I kind of grew up wanting to do either like science or language. And then I ended up doing science because I got to the end of like studying classics, Latin and Greek. And then I just realized I didn't have any more questions, but it was also April of my senior year. And so I needed to find something else to do real fast. So I ended up in a post-bac. And at the time, before I rotated in like cancer labs, but I had taken a microbiology course in my junior year that probably got me really interested in microorganisms in general. And just thinking about how something so small, both physically and when we talk about viruses, like the actual genome size can overtake something as large as us. And so that post-bac program was at the University of Chicago. And that's where I landed in the lab of Glenn Randall, who studied at the time hepatitis C virus, and had a really great time doing that. So much so that I applied to the University of Graduate School and stayed in his lab for my PhD, where I worked mostly on dengue. So.
Okay, cool. And had you been exposed to sort of science earlier on in like middle school, high school through your family, or was it just something that kind of came up later?
Yeah, it definitely came up late. Well, I think it was always there, but it definitely wasn't my family. So my parents are career Navy enlisted people. And so all they really wanted us to do was just like, my brother and I was like, A, don't assume the military will be the thing you do, and then B, figure out whatever it is that you want. And the only thing I can ever remember is I used to break a lot of stuff as a kid, like I guess most of us do. And so my dad told me I should go work at like Underwriter's Laboratory, where we could break things for a living. But aside from that, which was like when I was like 5 or 6, it was probably just what ended up being my own natural interest and what I was able to gravitate towards. My brother got all like the chemistry sets that I never played with and he never used. And I just ended up in science along the way. So.
And I guess, how did you find sort of your, the labs that you ended up going to? So how did you pick those labs in particular? What was it that sort of attracted you about those specific labs?
Yeah, so part of it is like the sloppiness of chance, but also like kind of also being like allowing myself that sloppiness, right? So after my post-bac, I realized that I wanted to do virology. And when I got there to Chicago, to do my post-bac, it was between either viruses or bacteria. And I just thought the question of something so small and navigating something so large was just even more pronounced for a virus person. So then I just kind of like found the right virus person to work with, and you got to do like little mini rotations. And Glenn and I had a really good like personal relationship, and then I enjoyed people in the rest of the lab. So it was a really good environment for you to land at and to like make mistakes and not feel like I'm the worst person. So that was really great. when I transitioned to graduate school, I wasn't dead set on staying in Glenn's lab, but as I popped around to some of the other bacteria labs in the department, because it was mostly bacteriology then, probably still is. There were some really interesting things, but there was nothing as interesting. I realized I wanted to be a virologist, like not a microbiologist, right? And then when I came to Ben's lab, I had seen him give a talk previously during my PhD, and he has a ton of energy, 1,000 ideas, and I was just like, I wonder if I study under him, if I can just like glimpse even a little bit of this for myself, right? Like, like he has like 1,000 ideas a minute. Can I just get like 10 and learn how he does that? So that was what made me excited about working with Ben and pursuing him for my postdoc.
Okay, cool. And I guess why don't you tell us a little bit about some of the research that you've been doing in your postdoc? Maybe some of the research that you were working on originally, and then maybe some of the research you've been doing on SARS, CoV-2, and COVID.
Yeah, so it's almost impossible to remember what I was doing before a year ago now, but largely it was just kind of studying a couple different individual antiviral factors. And so the favorite one from Ben was this protein DROSHA that he's described previously. So DROSHA is normally part of the microRNA processing machinery, normally resides in the nucleus, but during moments of stress, it'll translate to the cytoplasm. And it does this during viral infection. And this pathway seems to be distinct from other stress pathways that trigger this movement. And so we were really interested in that because once DROSHA comes out, it then has an antiviral activity against mostly positive RNA viruses. And so there's this, I really love the idea of things outside of interferon that are antiviral, right? And so just kind of cataloging these things. And along those same lines, he for about the first year of my postdoc, my goal was to like try to get a plant virus to replicate in human cells. And that also kind of really got me on this idea of like, we say like it doesn't work because like death of 1,000 cuts, right? This is like a transgenic boundary you're trying to cross, like whatever. But then you kind of wonder like, well, what are those 1,000 cuts, right? Like if I put you in like 293 T cells, which have no appreciable interferon response really, right? And you still can't replicate, like what is actually holding you back? And so that got me really interested in just kind of cataloging these factors that aren't interferon based, but also that are able to restrict viruses through their natural activity. That all got thrown away, like everything else everyone's done. And then, so about March last year, the end of March, the lab switched over completely to working on coronavirus. And in the beginning, I was just I don't have any I'm doing any animal work. And in the beginning, there was a lot of work being done in like ferret models, looking at the transcriptional profile of SARS-CoV-2 infected cells in the lab. And so I was just helping processing those samples and whatever random collaborations that would come through, because we were also one of the first places in New York to be able to use the coronavirus and have our facility up and running. And so around May, we got an email from our collaborator, Neville Sanjana, down at the New York Genome Center. He wanted to do a CRISPR screen. So that was awesome. There had been interest in doing it in the lab, but everyone was just crazy overwhelmed. And so they essentially just were like, if we make all the things, will you throw a virus on it? And I was like, that's fine. That's fine. That's exactly my bandwidth right now. And so the last, 7-8 months since then has been just, working through that screen, working on the hits, doing follow-up on those. And then probably around October is when things got kind of calm again, where I could start thinking about doing work of my own. And so some of that has continued with coronavirus, other collaborations, and then trying to understand the proteome of a replication complex and understand the formation of those things. It's kind of where we are now.
So I guess to follow up on some of the CRISPR screen, what was the, I guess, can you let us know what the goal of this particular CRISPR screen was? What were you looking for in doing it?
Yeah, I kind of wish you had thought about that first. So at first, the standard line with the CRISPR screen is you just want to see what's required, right? And then from there, especially in the pandemic sense, we want to pull out things that are druggable targets. And so we definitely did some of that, right? But the model we use, the ACE2A549s, while great at replicating the virus, then all you get is kind of like I think out of the top 36 hits, essentially like 33 of them were describing either the endocytosis of ACE2 or the recycling of it back to the plasma membrane, right? And so while that's useful biologically, it wasn't necessarily like what we wanted to know. But there's still kind of really good druggable hits. So there's a VPS34, which is PI3 kinase. We had quite a few drugs that were able to inhibit quite strongly, and drugs that have been published in other groups that fall in that similar pathway have also shown to have strong effects, and so that's been nice. But
okay, so I guess to follow up on that, is there a different way that you could use a CRISPR screen, do you think, to actually screen for more druggable targets, or?
Yeah, so
having gone through it once.
yeah. I mean, I would say a better way is a cell line that contains both the plasma membrane and endosomal pathway for SARS-CoV entry, right? So I have a particular bias. I think entry is the least interesting part of the virus life cycle. The virus gets in. I'm sure some of my friends will not be pleased with that, but so if you eliminate the restrictions of entry, then you're kind of really focused on what's happening for the virus to replicate and probably translate. And those tend to be will probably be could possibly be druggable targets, right? So that's how I would kind of redesign the screen if it was time.
Cool. And is that sort of the focus also of the proteome work that you're doing with the?
Yeah, so a little bit. It's like half useful, half nerdy. So when positive RNA viruses and pox viruses infect the cell, they create these beautiful replication compartments inside the cell. And this is something that's just enamored me ever since I first saw one in 2008 when I was working in Glenn's lab. And so I've always wanted a reason to be able to study them. And so now here's a super sexy virus that because of all the previous work in MHV and other beta coronaviruses and SARS and MERS, people know a lot about the viral components to forming these. And so now I can combine that information with getting kind of proteome of the cellular components that are there and asking what's important for the formation and what's important for the function, and what are these actual functions, because the function is kind of nebulous. But inside of that, one would also find all the different accessory factors the virus is recruiting in order to make this happen. And presumably some of those will be druggable targets. And then you can go down that pipeline and also ask, can we find a target that can be used therapeutically in the advent that the vaccines don't get the saturation that they need, or we have these mutant strains that pop up and evade immunity.
Right. Cool. So I guess more generally, what's been the most exciting moment in your career so far?
I think there have been two. They've been really small. And so the first was when I was rotating in graduate school in Glenn's lab. A postdoc, Kelly, she was doing a lot of live cell imaging. And so we had a lot of spinning disc confocal microscopes, so you could watch one channel and then switch over to the other. But for a while, the microscopy facility had a microscope with two cameras, so you could simultaneously view red and green. And I think we were just tracking endosomes and lysosomes, right? But I just remember like watching, watching the cell move and just be like, what is this? Right. Like, and just kind of giggling with joy. And then the other was when we were doing the CRISPR screen. So like the principle worked, right? Like we had screened cells before to see that which ones would die when we added the virus and after how long. But I guess I was still skeptical that I would actually like see anything. And so on day 6, when I looked under the microscope and I just saw like just little colonies of cells growing out, just like everything, there's just debris everywhere. And they just got these little cells just like popping around. I was like, this is just so silly. But yeah, so that was pretty nice.
Satisfying at the same time.
Yes.
Yeah. Well. And I guess on the converse side, what's been the most difficult thing you've had to overcome so far and as a scientist, and how did you overcome it?
There are a couple of things that come to my mind immediately. For one, I think, so I had a very long PhD. I had like a seven-year PhD. And so that does not instill confidence in oneself about like my ability to do science, especially science at a high level. And so kind of coming into my postdoc, and even while I was applying, I was just really nervous that like this was the end, like those 7 years of grad school were the end of my journey. And I really didn't have a lot of confidence, but then coming here, like moving to a different lab, being able to contribute both scientifically and intellectually, like really helped me kind of realize like, okay, I just wasn't good at the particular project that I had, and that's okay. But I'm not like necessarily like a bad scientist. And then the other one that's been interesting to get over time. So I'm Black. There aren't a lot of me in my institutions. It's not new. Most of the institutions I've been trained in from kindergarten to now have been majority white. And growing up like that, I remember teachers and peers telling me that I only got where I was because of the color of my skin, that I wasn't smart. So a lot of that you would internalize and carry with you. And I've thankfully never actually experienced it directly during my PhD or my postdoc time. But you still do end up like carrying this like weight on your shoulder of like, well, why are people reacting to me this way, right? And kind of just learning to not assume the worst, because the only person that puts work on is me, right? And it doesn't do anything. Like these people aren't thinking about that, and they're out here wondering why I'm carrying this burden. And so that took a while to just kind of like overcome, and it was just kind of the same process of realizing like, well, there are 1,000 different reasons why anything could be happening. You're the one giving the power to it being your identity. So how about just like playing that down a little bit?
Yeah. And is it helpful to reach out? Did you find sort of peers or mentors that helped you with that? Or was that something you had to do in yourself?
It's something I had to do by myself. I'm also a very go it alone type of person, so that could also help. But insofar as mentors, I think to date, I have like interacted with fewer Black faculty members than I have on one hand, right? I can count them. And the first one I had a substantial interaction with was in 2015, right, at a conference. And so it's a little too late along the path to wait to find someone to help you along. You have to kind of like do the work yourself up until then. And then you see someone else feeling like, okay, like we can get here, And then I've also had like colleagues and peers, and during graduate school, I was part of like the minority graduates, my minority graduate student council. And that was also really helpful, right? Because we all kind of had the same feeling of like, are we crazy?
Sort of related to that, if you had a chance to ask your older self one question, what would it be? So this would be you, say 60, 70, maybe in retirement. What would you want to know?
I wouldn't want to know anything specific, but I think what I would ask my future self is like, was it worth it? You know, like, as all of us know, like there's so much that you kind of give up to do science, like the long hours, like you miss family time, you miss friend times, people coming out of your lives because of this, right? And you just kind of like, and while you're doing the work, especially when you're like young, it's easy to say like, well, I'm doing it for like this greater cause, like I want to be a scientist. But then as time goes on, you start to like, I mean, does being a scientist have to cost all this, right? Right. And so just asking my future self, like, you know, was it worth it? Were we happy? Like, you know, I know you're not going to tell me anything that would like tell me to do something different, but you know, like I just kind of want to know, like, will I find the right balance? Will it have been worth it all in the end?
Yeah. I mean, I guess to follow up on that, how do you try and achieve balance now? As we know, it's, you know, science can be consuming, you know, like, and I do think that sort of good PIs are put on this pedestal where they just work constantly and that's all they do. And so like, how do you sort of balance, you know, being a good scientist, but also having a life?
Yeah. My balance is binge. And so I just binge one or the other. And I haven't really figured out how to do it on like a healthier way. But I will credit both Glenn, my PhD advisor, and Ben, my postdoc advisor, are people who, you know, obviously everyone wants you to work, right? But they also know that like broken people can't work. And so they encourage people to like take their time and like rest themselves and rest their minds. Glenn gave me one of the best pieces of advice ever. He was like, make sure all your friends aren't scientists because this is a weird profession and you should go meet normal people. And that's been really good because those friends don't understand why like we work the hours we do, right? And so they just also don't have patience for like, oh, I've got a 14-hour time point. They're like, no, just stop this, right? And that helps put in perspective, like, is this necessary to do, right? Or am I just doing this to say I was in the lab doing something? And so, you know, during those normal times, I try to find some balance like that. And then before the virus hit, like I played in like a flag football league that took me out of the lab every Saturday, right? And just active things to get away.
So I guess to follow up on that, how has the COVID-19 pandemic affected you as an individual? What's changed in the past year?
Yeah. Probably not a ton. So not a ton different than anyone else in the sciences, I guess. Like all my free time like was erased for a while. I'm happy to have a lot of that back though. You know, it's been interesting to compare myself to my non-science friends who like sit at home all day now, and where I still get to come to work and the joy that that brings me, not being stuck inside an apartment. I think definitely in the summer, I biked around a lot more than I would normally. So I wasn't super keen on taking the subway here in the city. So I would just bike around, and then that got me into doing like longer 40, 50, 60-mile rides with some friends, which is never a thing I thought I would do. But it was actually a lot of fun and a thing I hope to like can do in the future, you know, when it's warm. I don't really do this in the winter. But yeah, so a little bit of things like that.
And I guess as a virologist and someone actually who works on SARS, how do you make decisions about, and how have you been doing this in the past year, about keeping yourself, your family, and people safe, essentially, or talking about vaccines or not? How do you deal with that?
Yeah, I'm definitely very bullish on like talking about vaccines. In the summer, I was at a park with a couple of friends, And inside the group, it came up that half of them weren't going to take the vaccine. And I just like went blind with rage. And have since converted them all to vaccine adoptees. And I really do, like, especially in this, like when people don't understand the science, I'm very happy to take an active role in helping them like understand, right? Like I don't ever really want to have a large platform where I'm talking to 1,000 people, right? But like on these small intimate settings where you get to know a person, you get to understand their frustrations and the things that are making them hesitant, like being able to slowly walk through some of those and unpack them. Some people have wild conspiracy theories that you'll never unpack, and just please stop talking to me about this. And then for like making decisions on what to do, I think one of the things that was kind of like really done poorly was giving people the tools to accurately assess risk, right? And obviously anything you do that is not staying inside your house is like increased risk, right? But I often help my friends like through decision trees on like what they're doing, where they're going, and all the things that they should take into consideration. And then for them to know, like, yes, you can do a thing, right? But you should understand what might happen if you do that, right? And then just accept those consequences and make your choice based on those.
Yeah. What about, so as sort of for the vaccines, there's obviously a very a history, sort of like the Black community in vaccines. How does that sort of affect the way that you think or talk about it? So it's sort of I think not a unique, but a different perspective, obviously, because there's been this historical issues, obviously, with vaccination or public health campaigns. So how do you deal with that?
Yeah, it's like, it's one of these things that I know, but even still, every time I'm confronted with it, my mind is blown, right? Like between family members and close friends who are like, well, what about Tuskegee? And I'm just like, you can't always say Tuskegee. And so then I just really try to have a very a much longer conversation, right? And I also I don't try to like tell them that those fears are silly, right? Because they aren't, right? Because even today you can look into disparity data, right? And Black people have worse outcomes. And so the systemic problems still exist, and there's still an issue. What I think I was telling my friends who brought this up to me during the summer was, this is like, if I provide you the vaccine, and this is the best way to get out of this, and this virus is causing, more damage in Black and Brown communities, it's actually really important for these communities to take this vaccine, because otherwise you're just like, your historical fear, which is entirely grounded, is now perpetuating a larger damage cycle, right? And so you kind of have like, you have two options, right? You can get on board, or you can't, and then allow this thing to continue to run through your community and do tremendous damage, right? And the fact of the matter is, a lot of the trials were aggressive and recruiting minorities so they can have a sample size, and people were thinking about this, right? So I also try to communicate that like this isn't a blind spot, right? And that inside the data, there's nothing wonky happening with people of color, right?
So have you guys been offered vaccines there at your institution?
We have. So I was part of the Pfizer trial. And so I originally received a placebo, and then once it became licensed, we were able to get it through the trial. But shortly thereafter, around the same time, really, Mount Sinai itself was distributing vaccines. And because pretty much everyone in the lab works on the virus, we were all able to get the vaccine. I think within 2 weeks, the whole lab, which is like maybe 14 of us, will have all had both doses of the vaccine. And so that feels really nice. Yeah.
Yeah. Yeah. Well, thank you a lot for the volunteering. Yeah. People like you that made it possible. Having been vaccinated, how does that make you feel?
I really try to like pretend like I haven't been. It doesn't always work. But like you do feel like a little bit more relieved, right? Because like now I know the harm is going to be directly to me. And several of the friends that I see either already had it or have also been vaccinated. So like our little pod is safer than it was a year ago. But I still remind myself to think, and especially now, even more so about the other people I come into contact with, right? Because it's unclear and probably unlikely that having an antibody response will stop an initial infection anyways. You might still be able to be infected, transmit for a couple of days, but you just won't get severe disease or disease at all, right? But you might still be able to infect other people. So just kind of keeping that in mind, where I am now kind of like the worst case scenario out there, just like a person who could be like a walking time bomb, just like, spreading the virus to everyone.
Yeah. Great. Well, thanks so much for talking with us. Any last messages for our listeners? Any thoughts about the future of the COVID-19 pandemic? Where do you see us going in the next couple of months?
I mean, the vaccine rollout has been getting better, so it makes me more hopeful than I was 3 weeks ago for 1,000 different reasons. So I would just encourage people to get their vaccines, and maintain, keep wearing your mask, keep social distancing in the meantime. A friend of mine just learned that he wouldn't go back to work until September. And he's like, all I have is just the 3 rooms in my apartment. I'm going crazy. It's also winter. You can't go outside. So finding some way to really find a way to take care of yourself and find a small pot of people that you can still socialize with is probably even important, even more so now in the winter and as this thing gets longer and longer. So,
yeah. All right. Thank you. Tristan's work over the past year has focused on the use of CRISPR entry screens and proteomics of the replication complex to identify new, potentially druggable targets to prevent or treat COVID-19. In addition, he talks with friends and family to evaluate the risk and encourage uptake of COVID-19 vaccines. This has been Let's Meet the Virologists, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcasts, or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologists@gmail.com.